last set. The next set of notes is going to be endocrine system, which we will do. I don't know if we'll get started on it today, but even if we do, it'll just be a little bit. And then we're going to do it on Monday, and then you guys are going to work on it over spring break at least a little bit, or at least you're going to feel guilty about not working on it over spring break, which is what might happen. Okay, so we're going to do the autonomic nervous system today, and there is some anatomy review there for you. And we didn't start on this last time, did we? In this class? Okay, so first thing is big picture sort of intro to the nervous system as a whole. We've been talking about it, so it's not like we don't know this stuff, but the nervous system is basically going to be one of your body's two major control and communication systems, and the other one we're going to do right after this, which is the endocrine system. Nervous system tends to be really quick on and really quick off, and the endocrine system tends to be slow on and slow off, but they work together, and primarily their goal is to maintain homeostasis, primarily. But really the nervous system is going to do three things for you, and we've been talking about it because the last set of notes was really about the somatic nervous system and how it controls skeletal muscle. But the nervous system is really going to do three things for you. And if you want to think about what it does, think about putting your hand on a hot stove and then taking care of that problem. Because if you put your hand on a hot stove, you've got the information in, right? That's sensation. You got to process that information in the central nervous system. And that processing is called integration. And then that still hasn't solved the problem. Now we have to have information out, which will cause me to move my hand, right? Okay. So the information in is sensation. The information processing, so put your hand on a hot stove, the sensation comes in, and then the processing or integration that goes on in the central nervous system goes like this. Hot, which is like identifying what you've experienced. And then compare it. How hot? Like hotter than normal? Yeah. Identification and then assessment, which is too damn hot, right? But you still haven't fixed it. So the next part is to send the information out that causes you to move your hand, right? Okay, so the part of the nervous system we're going to be focusing on today is primarily going to be that third part, which is response, because we're going to be talking about the autonomic nervous system, which is specifically part of the motor division of the nervous system. And so we already know that neurons are special in their capacity to receive any kind of input, whether it's touch, temperature, pressure, pain, light, or whatever. And because of the fact that they have voltage-gated channels, they can convert it into an electrical stimulus. And then by virtue of the fact that they actually can release neurotransmitters, they can actually cause that to occur on other cells. So what we're going to be doing today is we're going to do a little bit of the functional organization of the peripheral nervous system. And you should have done this in anatomy, so this is something that we shouldn't have to go through slowly. We should be able to go through it relatively quickly. But since people have trouble with retention here, I'm going to make sure that you guys can do it. Okay, so when we're talking about central nervous system and peripheral nervous system, some questions for you. First question is this. Is the communication between the CNS and the PNS one-way or two-way? It's two-way. Do you agree? Yeah? Okay. So this is the same thing that I do in anatomy, and we're going to use it to start out here. If I had to make one of those arrows fit the word sensory, which arrow would it be? Which arrow, top one or bottom one, corresponds to sensory? I'm going to wait for you. I know two of you know what I'm talking about, but I'm going to wait for the rest of you. It's the top one, right? Okay, what's the ferent word for sensory? Afferent, exactly. Okay, so in phys, just so you know, you're far more likely to use the ferent words than you are to use the word sensory and motor. Okay, so you're going to have to get used to them if you weren't before. And so that's afferent, and then this word would this arrow would correspond to what word? Efferent, exactly, which is also motor. And so what's going to happen is, again, hand on a hot stove, the information comes in the afferent division of the peripheral nervous system and then gets processed or integrated. So afferent nervous system integration goes on, and then the command to move your hand comes out the efferent division of the nervous system. Okay? So just to make sure that you can really understand what we're talking about when we say we're going to do the autonomic nervous system, we're going to divide this and make sure you've got it. So the peripheral nervous system is not doing any of the integration, really, to speak of, right? So if you look at peripheral nervous system functionally, it's really going to be doing either sensation or motor activity, right? Either afferent or efferent activity, because it's not doing integration. So of the 3 steps, information in, information process, information out, it's doing the 2 on the edges, right? Information in, information out. All right, so with the peripheral nervous system, we're going to further subdivide it, and you've done this before. The afferent division, we're not going to be concentrating on today. The afferent division gathers information from sensory receptors and sends it to the central nervous system. You have all kinds of sensory receptors: touch, temperature, pressure, pain, proprioception, and you've got vision, taste, hearing, all of that business. We're not going to concentrate on that today. Where we're going to concentrate today is on this part, and this is the efferent division. And specifically, we have been talking about the efferent division in the whole last set of notes, because the efferent division has a subdivision, and it goes like this. The somatic motor division of the efferent nervous system stimulates what kind of tissue? Only one.
Muscle.
Be more specific. Skeletal muscle, exactly. So the other way of saying that is that skeletal muscle is the effector. Muscle effector. And so we've been talking about this one, right? We know what neurotransmitters we used. We know how skeletal muscle contraction occurs. So what we're going to do now is to fill in the other part of the efferent nervous system, which is over here, which is called the autonomic motor, or sometimes the visceral motor. Okay? And it stimulates the other stimulatable tissues. What other tissues can you control, although not voluntarily? Smooth, cardiac, and everybody forgets, yeah, glands. So this one goes to 3 effectors: smooth muscle, cardiac muscle, and glands. The other way of saying that is that these are the effectors. Go ahead.
Or basically, it's because when I think of glands, I think of just organs, but that just shoot out hormones.
Yeah, but many of them are controlled by this part of the nervous system. That's what we're talking about. So, for instance, salivary gland secretion is controlled by this branch of the nervous system. Okay. So the nervous system is deciding, generally subconsciously, when to secrete salivary amylase or when to salivate, for instance. So, yeah, we're going to see examples of that. Okay, but of course this one is more complicated because it has a further subdivision, right? The autonomic motor division has a further subdivision, and that is this, and that is that it is further subdivided into the sympathetic branch, which is often referred to as fight or flight, but it's really fight, flight, and exercise. And it's going to the same organs as the parasympathetic branch. And the parasympathetic branch is usually referred to as feed or breathe and rest and digest or whatever. I usually refer to it as feed or breathe. Okay, so immediately we notice that this is different because the skeletal muscle basically is only going to have one hookup. So stick with me for a second. That means with skeletal muscle, I either stimulate it and get it closer to contraction or just leave it alone, okay? But what I'm telling you is with cardiac muscle, I have 2 possibilities. I can actively increase its activity or I can actively decrease its activity, okay? I've got 2 different branches. And so you guys have heard of the fight or flight division before, so let's just talk about it generally and then we'll get into the specifics of how it works. So basically, with these effector organs, we are going to have what we call dual innervation. Dual innervation means the nervous system is hooked up 2 different ways to the heart. And I'm going to use it in 2 different ways, so stick with me for a second. So what's going to happen is right now, you're relatively chilled out, your quiz is over, your test isn't until next week. You're not running around the building, right? So if I'm thinking about what's going to be happening to my heart, what I'm going to be doing is I'm going to be using one branch, this one, to actively inhibit or slow down what the heart is doing. I'm going to be like putting my foot on it, right? Saying, I want you to slow down even below your pacemaker potential. I want you to be even slower than that. But then what happens is the tiger walks into the room, right? The sympathetic nervous system is going to go, I got this. And the parasympathetic nervous system is going to go, you got this, I'm out. Because the sympathetic nervous system is now going to actively increase the activity of the organ. So instead of just having one hookup like the somatic nervous system does, and either I stimulate skeletal muscle or I leave it alone, I can actively turn it up or I can actively turn it down. Okay? That's what the autonomic nervous system does. Okay? Has two sets of hookups, therefore two different kinds of physiological possibilities. You guys understand that? Is that good so far? No? Yes? No? I need you to give me feedback today. Okay, go ahead.
Okay, so when I'm looking at it, just terminology-wise, para means like close by or something like that. So sympathetic doesn't have the para in front. Is this also due to like location?
No, it's not due to anatomy.
Okay.
That anatomical location doesn't really make much of a difference here. Okay, so so we've already talked about the afferent division, and we're really not going to concentrate on that. We're going to concentrate on the efferent division. The efferent division is going to be subdivided like this. The efferent division is primarily going to be communicating with these effectors, and it's going to be carrying information away from the central nervous system, right? Ultimately ending up in muscles and glands. And now we're going to start building a little bit more physiology and neurotransmitters into it. So this is just the basic organization of the terminology that we're going to be using. I actually expect you to have already learned this, okay? I expect you to have come in with that. That is not new information as far as I'm concerned. So, but what probably will be new information is this part. So what I'm going to do now is I'm going to take and break down, sort of put the anatomy together with the physiology of the cells of the entire efferent nervous system. So this is going to be the entire efferent nervous system. And I'm going to further subdivide it into the somatic and the autonomic. Okay? And then I'm going to tell the parts of the story that you already know and then fill in the parts that you don't. Crap. Okay, so with the somatic, if I want to say what is the effector, what is the effector for the somatic nervous system? It is skeletal muscle, exactly. Okay, so you can even recognize a bad picture of skeletal muscle, right? Which is the only kind of picture I draw, okay? So the effector is skeletal muscle, everybody agree with that? Okay, and the effectors for the autonomic nervous system are cardiac muscle, smooth muscle, and glands. I'm just going to draw the one that's easiest to recognize, okay? And that's going to be this guy right here, which is what? Cardiac muscle, right? That's cardiac muscle. Okay, so now we're going to fill in this part of the story because we already know it. So now I'm going to say I want to understand the arrangement of how the neurons communicate with the skeletal muscle fiber. And you actually already know that. I've got the central nervous system and the peripheral nervous system. Okay, so the question I'm going to answer right now is if I am trying to make the muscles in my hand contract, where is the neuron cell body that is actually going to cause that to occur? The neuron is going to be up here in the cervical region of my spinal cord. And the only thing that's going to be running through the peripheral nervous system is actually the axons, right? So, yeah? Okay. So what you're going to have is the neuron, which is a somatic motor neuron, right? It's going to be in the central nervous system. And the only thing that's going to run through the PNS is the axon. And then a couple of things that you already know. What neurotransmitter is always used here? It's always acetylcholine. And what is the effect or response? Excitation or inhibition?
Excitation.
It's always excitation. Okay, that's all stuff that you know. And we're using this as a comparison contrast because the autonomic nervous system is more complicated. Okay, but this is stuff that you already know, right? Okay, let's correspond it to what's in your notes. So with the somatic division, The somatic motor neurons control skeletal muscle, or skeletal muscle is the effector. This is usually controlled voluntarily because, just so you know, that neuron right there is ultimately hooked up to your cerebral cortex, which is the voluntary portion of your brain, right? And so that means that generally you can control skeletal muscle voluntarily. Not always, but generally you can. And so the efferent route right here from the CNS to the skeletal muscle fiber is simple. The somatic motor neuron cell body is in the brain or the spinal cord. The axon goes through the PNS to synapse the skeletal muscle fiber at the neuromuscular junction. So there's only ever one synapse. It's really easy on its way there. But we're going to compare that to the autonomic nervous system. The autonomic nervous system, also abbreviated very commonly the ANS, is involuntary, which means it probably won't surprise you that this is not hooked up to your cerebral cortex. It's hooked up to older involuntary portions of your brain like your pons and your medulla oblongata and your hypothalamus. Does that make sense? So this is involuntarily controlled. And ultimately it is going to end up at 3 possible effectors: cardiac muscle, smooth muscle, and glands. We didn't draw all 3 there, but they can go to all 3. So since it's not hooked up to the voluntary divisions of your nervous system, the control is involuntary, right? Okay, so now what we need to do is we need to talk about how you are going to get there. And the way that you are going to get there is just a little bit more complicated. It's not super bad. And so basically we're going to build this, the same thing. So here's what's going to happen with the autonomic nervous system. Again, we always start in the central nervous system, right? You have to start everything there. So there is a neuron that is in the central nervous system, the first one. And then what's going to happen is it's going to make a pit stop on its way to synapse with another neuron. And then that neuron is going to go to the effector. Okay? So we're going to have some parts of the story that we have to tell. First off, anatomically, do you remember what you call it when you have a blob of neuron cell bodies in the peripheral nervous system? Yeah, it's called a ganglion. Exactly. So this is called an autonomic, because autonomic, ganglion. Okay, so just a little bit more information and then we'll be able to back up and put some more detail to it. So now, just based on the anatomy and the physiology that you already know, if I asked you to, you could probably very easily tell me which one of these 2 neurons on the bottom could be termed preganglionic and which one could be termed postganglionic. Yeah? Okay? Everybody agree that this one's preganglionic? And this one's postganglionic? Good? Okay, so now there's some physiology that we're going to have to fill in. Notice that we're going to have 2 different synapses, right? So we're going to have to figure out what neurotransmitter is used here, right? And what the effect is. And then what neurotransmitter is used here and what the effect is. Does that make sense? So that's the parts of the story that we're going to have to tell. Okay. So, but of course there's not just one division of the autonomic nervous system. There's sympathetic and parasympathetic. Let me see if this makes conceptual sense before we go any further. If the sympathetic, for instance, turns up the heart rate, right, and the parasympathetic turns down the heart rate, does it make sense to you that they can't do it by releasing the same neurotransmitter, right? So what you're going to have to do is you're going to have to subdivide the autonomic nervous system into the sympathetic and the parasympathetic to get some detail in there. Good? Okay, so does that make conceptual sense to everybody? That you can't actually do 2 different things at the same organ by releasing the same neurotransmitter. So that's what we're going to do in just a minute. We're going to subdivide that into the sympathetic and the parasympathetic. Okay. But before we do, we're going to put a little bit of functionality into the sympathetic and the parasympathetic so you'll be able to predict a little better. Okay. So there are 2 divisions of the autonomic nervous system. And so, like I said, most autonomic effector organs are dually innervated. And so what I want to show you is this. It's not as bad as it looks, and hopefully you looked at it in anatomy. So this looks horrible. but it's not horrible. It's really not, I swear. So what I'm saying is, let's just concentrate on the heart because everybody can find the heart, right? Okay. So what I'm saying is that there's two separate sets of wiring, nervous system wiring, and specifically motor wiring going to the heart that I use under two different circumstances. So the sympathetic nervous system is typically referred to as fight or flight, right? And the parasympathetic nervous system is typically referred to as feed or breed. So now I want to put together both the anatomical root and sort of a little bit of the functionality of what's going to go on right now in this room, okay? Right now in this room, your level of stress is not as high as it could be, although if I keep using these words, she's going to throw something at me. Sympathetic, parasympathetic, preganglionic, postganglionic, excitatory, inhibitory. It's not as high as it could be. So which branch do you think is controlling your heart right now? Parasympathetic. Okay, so I want to sort of trace the anatomical root to what is going on with your heart right now, okay? So in tracing the anatomical route to get to the heart, what's going to happen is the parasympathetic nervous system generally goes through a few of the cranial nerves and then some of the sacral nerves. But if we're trying to end up at the heart, what we are going to do is, follow me, go through cranial nerve number 10 to get to the heart. Do you remember what number 10 is? Vagus nerve. Okay, so now what I'm telling you is this is what's going on right now. Cranial nerve number 10, the vagus nerve, which comes out near the base of the brain, right? Is actually, there's two of them, there's a pair of vagus nerves. They are actually going down and they are actually releasing their neurotransmitter at the cardiac muscle right now, which is actively inhibiting the cardiac muscle from going any higher, right? That's what's going on right now, okay? And it's going via that anatomical root, cranial nerve number 10, okay? But now a tiger walks into the room. And we are all screwed if they figure out how to open doors. We can definitely agree with that, but they can't, luckily. But a tiger walks into the room. And so now I want to know, what is the anatomical route to the heart now, okay? So what's going to happen with the parasympathetic nervous system right now is that vagus nerve is going to go, I'm out, you got this, right? And so the vagus nerve is no longer going to be trying to actively inhibit the heart. And I am going to try to get the sympathetic nervous system to excite the heart, but please note that it actually goes through a different anatomical route, right? It goes from the brain down to the thoracic region of the spinal cord, right? And then over and up to the heart, okay? And it's going to release a different neurotransmitter because it wants a different activity to go on the heart, right? So when you get stressed out, when you almost get in a car accident, when a tiger tries to eat you, when a rhino tries to run you down, I don't know what the hell happens in your life. Somebody tries to hit you upside the head with a frying pan. What happens is it goes down to the thoracic region of the heart, down to the thoracic region of the spinal cord and up to the heart, and then it releases a different neurotransmitter. So that's what we're talking about dual innervation, right? There's two different anatomical and physiological hookups that we have here. Okay, so the sympathetic activates fight or flight and exercise responses of cardiac muscle, smooth muscle, and glands. And the parasympathetic activates feed or breed responses. And generally what they're going to do is they're going to work opposite one another. So the good part is that if you can figure out what the sympathetic nervous system does to the heart, then you don't have to memorize what the parasympathetic nervous system does to the heart, right? Because they generally work in opposition to one another. Is that good? Yeah? So if you know that the sympathetic increases and that makes sense to you, you don't have to memorize the other one. Just like, oh, then the other one does the other thing, okay? So these two, here is the mistake that a lot of students make. A lot of students make the mistake of thinking that the sympathetic nervous system actually increases the activity of all organs that it's attached to, and the parasympathetic decreases. But that's not true, because the way that you approach this is thinking about resource allocation, okay? So we can probably all agree that you have a finite amount of blood, finite amount of oxygen at any given time, and a finite amount of ATP at any given time. Good? So if a tiger walks into the room, do you really want to increase the activity of all of these organs? No, you want to increase the activity of any organ that could help with the situation and decrease the activity of any organ that would not help with the situation. So you already understand the fact that when the tiger walks in the room, the sympathetic nervous system will increase cardiac muscle activity. But what do you think the sympathetic nervous system should do to the smooth muscle in the wall of the uterus? decrease it, right? It's resource allocation. You have a finite amount of blood and resources, and you don't even care about your uterus at that moment. As a matter of fact, I would throw it at the tiger if it would get me the rest of me out of there, right? You're like, just take it. I'm done with it anyway. So you need to turn down some things and up other things. And that is, you can memorize it if you want to, but if you can just logically think through resource allocation, you're going to get it right most of the time. There's only a couple that are counterintuitive. And I'll tell you those a little bit later. Is that cool? Okay, so sympathetic and parasympathetic are going to work in opposition to one another generally. So before we get back to filling in the neurotransmitters and talking about how they're going to cause different activities on the same organ, we're going to try to get to the point that we have good predictive capacity to figure out what the sympathetic should do to an organ and what the parasympathetic should do to an organ. But before we even do that, I want to make sure that we reinforce terminology. Because, like I said, the concepts in this set of notes are usually kind of intuitive. Like, you're like, yeah, that makes sense, I've got it. But the terminology completely throws people. So what I want is for you to get comfortable enough when I say autonomic nervous system, you can immediately tell me whether I'm talking about afferent or efferent. If I say autonomic nervous system, am I talking about afferent or efferent? Efferent, yes. And if I say autonomic nervous system, is it part of the CNS or the PNS? What is it? Part of the CNS or the PNS? Yeah, it's part of the peripheral nervous system, exactly. And it is mainly under control of the hypothalamus, the pons, the medulla oblongata. It is generally involuntary and automatic. Every once in a while, like if you've got like a yogi or a martial artist or a CIA operative, they can, for instance, train the cerebral cortex to override the functionality. And we'll talk about that at the end, assuming we have time today. So what we're going to do right now is a tour of some of the functionality of the sympathetic and the parasympathetic nervous system. This is not meant to be a complete list, but what it is supposed to be is sort of get you to the point that you're feeling comfortable with your capacity to predict what the sympathetic and the parasympathetic are going to do to each one of them. So in each situation, we have an organ, a tissue, and then we have what the sympathetic does and the parasympathetic. And each time you're going to have to figure out whether what it is doing is representing excitation of the tissue or inhibition of the tissue. And as far as the tissues go, we've only got 3 choices because the effector organs for the autonomic nervous system are 3 things. What are they? Smooth muscle, cardiac muscle, and glands. So in every single one of them, you fill in one of those 3 at least, okay? So let's do the first one together and you'll see what we're talking about. Ideally, what's going to happen by the time we get finished doing this table is you will prove to yourself that A, you have a pretty good predictive capacity for what the sympathetic and the parasympathetic is going to do, and B, that the sympathetic does not excite every tissue and the parasympathetic does not inhibit every tissue. So first thing is the eye, and specifically what we're going to be doing is try to control the diameter of the pupil, okay? And what tissue of the 3 would you guess is responsible for controlling the diameter of the pupil? It has to be smooth muscle, right? Because the other 2 don't make a damn bit of sense. If you say them out loud, you're like, no. But in order for you to figure out whether you are going to excite it or inhibit it, I have to show you the anatomy of how that works. I don't know if you'll remember it. So here's the pupil of the eye. And there are smooth muscle fibers that are arranged like flower petals around the smooth muscle, around the pupil of the eye. Okay? And this is sort of an oversimplification, but think about it this way. Here's the origin and here's the insertion. And now my question to you is, how do I cause pupil dilation? Do I excite those smooth muscle fibers and contract them, or inhibit them and relax them to dilate the pupil? Which one? Go ahead, Ryan.
Excited.
Yeah, you excite it, because if you excite it, it's actually going to pull the hole open. Do you see? Because of the anatomy. Is that good? Okay. So now, before we go any further, so this is smooth muscle. The sympathetic nervous system dilates the pupil. Can anybody put the logic together with me of why that might be worth it? So you can see every single part on the tiger is about to eat you, right? You can see every single part on Smaug the dragon, so you can find the one scale where you can see, nerd. So you can find the one scale where you can shoot your arrow, right? So you're going to see as much detail as possible, and that's why it's worth the money, right? Worth the ATP. Okay, so it dilates the pupil, and we decided that that represented excitation and contraction or inhibition and relaxation. Excitation and contraction, right? So then you don't have to memorize what the other one does, right? Because if the sympathetic nervous system dilates it, the parasympathetic nervous system will constrict it, okay? How do you think you would allow for constriction? Excitation or inhibition? Inhibition, exactly, just based on that anatomy. Good? Okay, so now, salivary gland. What kind of tissue are you dealing with? Gland. Good job. Yes, gland. So the sympathetic nervous system decreases salivary gland secretion. Before we go any further, how does that make sense? Yeah, what does saliva do for you? What does it do? Yeah, it's digestion of carbs, right? And so if a tiger is about to eat me and I have just eaten an Egg McMuffin, he can digest my damn carbs, my Egg McMuffin for himself, right? So salivary gland secretion cuts off, right? So does that represent, that one's pretty easy, excitation or inhibition of that gland? Inhibition, exactly. So parasympathetic nervous system increases secretion by doing what? Excitation or inhibition? Excitation, good. Okay, so now the next one, the lungs, specifically the diameter of the bronchioles. We can't control the diaphragm with the autonomic nervous system because that's skeletal muscle, but we can control the diameter of the bronchioles, right? And so what of the three tissues do you think is responsible for controlling the diameter of the bronchioles? Smooth muscle. But the anatomy is different. So I'm going to show you the anatomy, and then you can figure out how the sympathetic nervous system would allow for dilation of the bronchioles. Before we go any further, does it make sense to you that the sympathetic nervous system would dilate the bronchioles? And if so, how, in your words? Why? Why would the sympathetic nervous system dilate the bronchioles? More air, more oxygen, right? More cellular respiration, more ATP. Okay, so in order to answer whether that represents excitation or inhibition, I have to show you the anatomy. So here's the bronchiole, a cross-section of the bronchiole. And here is how the smooth muscle fibers are arranged around the bronchiole. And so the question is, if I stimulate these, will they constrict the bronchiole or dilate the bronchiole? If I stimulate them, they will constrict the bronchiole. So how do I allow for bronchiole dilation? Excitation or inhibition? Inhibition. Yeah, inhibition allows for dilation. Okay? And that means that the parasympathetic nervous system will constrict them back. Good? Okay, so next one is very, very easy. The heart, what kind of tissue? Cardiac muscle, exactly. And the sympathetic nervous system increases heart rate for relatively obvious reasons, which represents excitation or inhibition. Excitation. And the parasympathetic nervous system decreases heart rate, which represents inhibition. Okay, so now the next one. Stomach and intestines. The stomach and intestines do two things related to autonomic function. They have motility, which is what kind of tissue? Smooth muscle, and secretion, which is glands, exactly. So motility and secretion, so you're going to have both of those tissues there. So the sympathetic nervous system, how does it make sense that the sympathetic nervous system decreases both motility and secretion? Why does that make sense? Right, because that is ATP that you want to spend elsewhere, right? You do not want to spend it on digestion at that particular moment. So you decrease both motility and secretion, and that represents what? Excitation or inhibition? Inhibition, exactly. And the parasympathetic nervous system increases motility and secretion, which represents excitation. Okay, so the next two are the hard ones. You can do them, though. Just make sure that you get them before we go on. Okay, now we're going to talk about blood flow. Okay, so when you are controlling sort of regional blood flow and a tiger walks into the room, do you agree that you need more blood in some places and less blood in other places, right? Okay, so we are not going to, for instance, vasodilate to everywhere or vasoconstrict to everywhere, okay? But we are going to control the diameter of arteries and arterioles that are supplying certain places. So I'm going to give you the two extremes that we've got here. So first we're going to talk about the arteries and arterioles that are supplying the skin, right? And the viscera. Viscera usually means stuff in the abdominopelvic cavity, okay? So what tissue, first off, is going to control the diameter of arteries and arterioles supplying anything? Smooth muscle, exactly. Okay, so stick with me here. Do I need to increase or decrease blood flow to the skin and the viscera with sympathetic stimulation? Skin and viscera. Decrease blood flow to skin and viscera. Does that make sense? You've got 5 liters of blood. The vast majority of it you want to go to the brain, heart, lungs, and skeletal muscle, right? The rest of it, you don't really care whether it's going to the wall of your stomach, the wall of your uterus, the wall of your urinary bladder right now. Okay, so what's going to happen is the sympathetic nervous system is going to constrict the arteries and arterioles leading to the skin and viscera, and all I have to do is tell you that it's got the same anatomy as the bronchioles, and you can figure out how you would cause constriction, excitation or inhibition. How do you cause constriction with these smooth muscle arranged this way? Nope, not inhibition. Excitation. If you excite these, then they are going to contract and decrease the diameter of the lumen, okay? So excitation. Just think about inhibiting them. If you inhibit them, they'll relax and then the lumen gets bigger in that situation. Okay, so now here's the exception to the dual innervation rule that we've been talking about. You don't have dual innervation anywhere in the body of the smooth muscle in the walls of your blood vessels. Here is how I cause more or less constriction in this place. It's just more sympathetic stimulation causes more constriction here. Less sympathetic stimulation causes less constriction, which is dilation, comparatively speaking, okay? So you don't have dual innervation of smooth muscle in the walls of your blood vessels. It's just more and less sympathetic stimulation. Okay, so now keep that in your mind, and I'm going to tell you a different example of blood vessels. So now I want to talk about a different set of arteries. Same tissue, right? Now I'm going to be talking about arteries and arterioles that are supplying skeletal muscle, okay? Do you agree that it's going to still be smooth muscle that will control their diameter? Still going to be smooth muscle. But now I want you to think about what should happen to the arteries and arterioles supplying skeletal muscle with sympathetic stimulation. Should they dilate or constrict? Do you agree that they should dilate? Yeah? Do I need more or less blood to skeletal muscle? I need more blood to skeletal muscle. Correct, right? So here's the next part of the question. Same anatomy, and it's even going to use the same neurotransmitter. But now I want to know, how would I cause dilation here? Would I excite it or inhibit it? I would inhibit it. Okay, so I'm going to inhibit the smooth muscle here, which allows for dilation and increases the blood flow to skeletal muscles. And this is where it gets to be a head scratcher, but you know the answer. How on earth can you actually have the same neurotransmitter cause 2 different responses? Different receptors, right? So it probably should make sense to you that the receptors in the wall of the arterioles leading to skeletal muscles respond differently to norepinephrine, for instance, than the receptors in the smooth muscle of the wall of the blood vessels leading to their skin. Okay? So that means even though I'm going to release the same thing, I'm going to respond 2 different ways to it. And that's going to be important. So same thing here, though. You have no parasympathetic innervation here as well. So here. So that means that, stick with me, that means that with more sympathetic stimulation, that's how you cause more dilation here. With less sympathetic stimulation, you cause less dilation, which is constriction. And these 2 respond differently just because of different receptors, that's all. Okay? All right. The last one is the adrenal medulla. So where are your adrenal glands? On top of the kidneys. And if you have a cortex and a medulla in any organ, where are they relative to one another? Cortex and a medulla? Yeah. So you have an adrenal cortex and an adrenal medulla. Where are they relative to one another? Medulla's on the inside, exactly. So this is the deepest part of the adrenal medulla, the adrenal gland. What kind of tissue are we talking about here? Yeah, it's a gland. Okay. So let me show you your adrenal glands if you don't remember where they are or what they're like. So here's the adrenal glands. There's this outer portion, and we'll talk about its function in the endocrine system, and there's this inner portion. The inner portion is the adrenal medulla. Now, the adrenal medulla is an interesting story because the adrenal medulla actually really puts together the nervous system, which is what we've been talking about, and the endocrine system, because when the sympathetic nervous system stimulates the adrenal medulla, it will secrete epinephrine, adrenaline, and norepinephrine, noradrenaline, into the bloodstream. Okay, so all of these things that I have been doing with epinephrine and norepinephrine. And now I'm going to dump epinephrine and norepinephrine into the bloodstream. So let's say it takes me more than 5 minutes to get away from the tiger. It takes me 30 minutes to get away from the tiger. I could either repeatedly release epinephrine and norepinephrine as a neurotransmitter, or would it be cheaper or more expensive to dump it into the bloodstream? It would be cheaper to dump it into the bloodstream. So what happens here is with sympathetic stimulation, you will actually zap your adrenal medulla and it will release a neurotransmitter-like substance into the bloodstream. So it will behave as a hormone. And what it's going to do is enhance the fight or flight response and prolong it as cheaply as it possibly can. And you have totally experienced this. I know that you have. So the last time somebody almost killed you on I-15, right? You almost died on I-15 and you saw your life passing before your eyes, right? So what happened? Well, let's look at it. You had a bunch of things that you noticed and some of them that you didn't. If you had looked in the mirror, you would have seen that your pupils got enormous, right? Sympathetic stimulation. Your salivary glands will shut down. Your mouth will get really dry. You probably didn't notice that your bronchioles dilated, but you might have noticed that your heart rate went up. Your stomach and intestines will kind of turn off. Your skin will get sheet white, right, as the blood is shunted away from your skin. And you probably didn't notice what happened with your skeletal muscles, but you send a whole bunch of blood to your skeletal muscles. In addition, if it's a big enough fight or flight response, you will dump adrenaline into your bloodstream. Epinephrine, norepinephrine into your bloodstream. And then what will happen is, let's say you figure out that you're not going to die. Are you going to calm down in 30 seconds? No. After it gets dumped into your bloodstream, then the fight or flight response is enhanced or prolonged until you break down that epinephrine and get it out of your bloodstream. A lot of that has to go through the liver in order for it to occur, or it gets broken down by the target cells, but it's not instantaneous, is it? Right? It takes you a little while. And you can experience that as a parent, or you can experience that as a spouse, or whatever. So if, for instance, you ever let your kids go to the toy store and get in full-blown flip-out mode because they want a toy that you're not going to get them, and then you're like, calm down. I said calm down. They can't calm down. They've got adrenaline pumping through their bloodstream, right? So the best that they can do is you can like put them someplace that they can throw a fit without damaging anyone or anything, right? So it takes a little while for it to get out of your system. Or if you, like, this is what I typically do with my husband. Well, not frequently, but I certainly almost do it a lot of times. So if you get to the point that you're about like ready to scrap with your significant other, and you're like, you've got like your argument all built up, and you're about to present your argument, and you're like, hey, Mike, I got to talk to you. I want to talk to you about this and this and this, and you did this thing. And then he says, oh, you're totally right, sweetheart. I'm really sorry. Then what do? You're like, you just keep going, right? Because you're like, adrenaline is already in there. And if you have the presence of mind and you remember you're a grown-ass woman, you're like, turn around and walk out until you can get it out of your system. Because it's not the wise part of you. It's the adrenaline that's just dumped into your system. So this makes it last longer and it makes it stronger. In addition, we will see in the next set of notes that it is not limited to those 3 target tissues, because now it's in your bloodstream and it can actually impact other tissues. And we will talk about its impact on other tissues. So that is the adrenal medulla. It also has no parasympathetic innervation because you don't really need it. You just don't stimulate it when you're not in fight or flight mode, and you do stimulate it when you're in fight or flight mode. Although not always, you don't stimulate it every single time. You're even slightly surprised. Okay, so now what we're going to do is we're going to go back to this, and we are actually going to separate it out and talk about the neurotransmitters that are involved in this story. So the Anatomic Organization of efferent neurons and synapses, which we've already done some of it. So the things that we've already done, and then we're going to separate it out. All right, so we've already done this part. This is basically what is written here is what we drew here. The somatic nervous system has one neuron that stretches from the CNS to the effector, right? Therefore, there's one synapse called the neuromuscular junction. The cell body is in the CNS. Is this true? The axon runs through the PNS. There are no ganglia on the way. The neurotransmitter is always the same. It's always what? Acetylcholine. The effect is always excitatory on that skeletal muscle fiber, and you should know this. The mechanism of excitation is generally to open what kind of channels? Sodium, and cause what kind of polarization? Depolarization, and those synapses would be what ergic? Cholinergic, exactly. So now all we have to do is separate this one out. So we already told the first part of the story. The autonomic is a 2-neuron chain on your way to the effector. The first neuron is considered preganglionic, and its cell body is in the CNS. The second neuron is considered postganglionic, and its cell body is actually in the ganglion, and it goes to the effector. There is one exception to this 2-neuron chain rule, and that's this little guy that we just looked at. So the exception to the 2-neuron chain rule in the autonomic nervous system is the adrenal medulla, because look, it's just weird. So here's the sympathetic. This is the preganglionic one. You guys with me? Okay. And this weird looking one is a highly modified neuron that's an anaxonic neuron. So when it releases its neurotransmitter-like substance, it gets dumped into the bloodstream instead of released as a neurotransmitter. So it's kind of an exception to the 2-neuron chain, only really it's just the second neuron is weird looking, really, and it just ends up dumping something. So the adrenal medulla actually dumps it into the bloodstream instead of at a synapse. It just dumps it into the bloodstream and in much higher concentration. Okay, so now what we have to do is we have to separate this out. So we're going to separate this out. We're going to continue this story and tell the other side of the story. So the whole thing is still going to be the autonomic nervous system, and we're going to separate it out into the sympathetic and the parasympathetic. Okay, and so we're going to again end up at the same effector, and let's just make it cardiac muscle. Could be smooth muscle or glands. So this is going to be the effector. And then the last column is going to be the effect. Okay, so the part of the story still goes CNS, PNS. So sympathetic and parasympathetic both start out with a preganglionic neuron. And then the sympathetic is going to find the second one in a ganglion. That's not different. And then it's going to go to the effector. So we'll figure out neurotransmitter, neurotransmitter effect here. And then here we're going to do the same thing. So the parasympathetic going to find the second one, and it's going to go to the same effector. Okay, your textbook, by the way, has the same thing that we're building right here, but until you're used to looking at it, it's a little overwhelming to see in class. So it's there, and it's drawn better than I draw. But I think usually it's more helpful to build it yourself at least once. Okay, so what we have to answer in each of those situations is neurotransmitter effect, right, in both of those locations. So here we go. The preganglionic neurons, you can find them, right? The preganglionic neurons in both the sympathetic and the parasympathetic nervous system release the same neurotransmitter here, and it is always acetylcholine. Acetylcholine here in the ganglion binds to cholinergic receptors, specifically they're nicotinic cholinergic receptors, and the response is the same in both situations. The response is excitation. What I mean by that is not that it excites the cardiac muscle at the end, but that the signal doesn't go halfway. It continues to go, right? So you're going to excite this one so that it's going to end up giving the message. You're going to excite this one so that it's going to end up giving the message. So now I want you to see that there is now a trend with the entire efferent nervous system before we go any further. do you see that the entire efferent nervous system, this is the whole thing, right, releases acetylcholine first, right? Acetylcholine, acetylcholine, acetylcholine. That's all the efferent nervous system. And when released first, acetylcholine is always excitatory. Do you see that? Gives you way less to memorize, right? So there isn't a second here, I agree, right? But it's still released first and it's excitatory. And when it's released here, right, here, it's excitatory as well. So acetylcholine is always released first in the entire efferent nervous system, and when released first, it is excitatory. Now, we know that it can't release the same neurotransmitter if it's going to have two different effects on the same tissue, right? So that's where it's going to differentiate, because the parasympathetic nervous system is going to release acetylcholine again, okay? But the sympathetic nervous system is not. The sympathetic nervous system is now going to release norepinephrine and or epinephrine. Okay, so really, guys, this is the only place that it really differs. Okay, it's right there. So here's my question for you. The effect. Now, these two are definitely not going to have the same effect, but can you predict the effect if you don't know the specific tissue? No, this is where you start using your logic, right? Your resource allocation logic. And you say, well, at cardiac muscle, epinephrine is going to be excitatory, right? And that means acetylcholine is going to be inhibitory, right? But if instead of cardiac muscle, we drew the smooth muscle in the wall of the digestive tract right there, then epinephrine would be what? Excitatory or inhibitory? Yes, inhibitory. And acetylcholine would be excitatory. Does that make sense? So what you do is you memorize all of this, but when you get to the effect, you use logic, right, to figure out what it's actually going to do. That's why we did that entire table, once we got to the point that we could actually sort of confirm what we were saying. So let's go back to the notes for just a second. For adrenal medulla, yeah, it's excitatory with the sympathetic nervous system. Okay, so neurotransmitters at autonomic neurons. Let's fill this in and make sure it corresponds with this. So the preganglionic neurons of both divisions, sympathetic and parasympathetic, are going to release acetylcholine. It's going to bind to nicotinic cholinergic receptors. The effect is always excitatory, and the mechanism of excitation is what you would think it is. It is generally to open what kind of channels and cause what kind of polarization. Sodium, D, exactly. This synapse right there in the ganglion would be what ergic? Cholinergic, exactly. And then where it differs is here. The postganglionic neurons. The postganglionic, and just make sure it corresponds with what we drew. The postganglionic neurons in the parasympathetic division will release acetylcholine again, which binds to, this time it's muscarinic cholinergic receptors. And then that synapse, this one right here, would be what ergic? Nope, this one. That one's still cholinergic, right? And the postganglionic neurons in the sympathetic nervous system right here release norepinephrine or epinephrine. And by the way, when you're releasing norepinephrine or epinephrine, if you're releasing it as a neurotransmitter, it's about a 4 or 5 to 1 ratio of nor to epi. And when you're releasing it as a hormone, it does the opposite. So norepinephrine is far more common. So norepinephrine is released here. And what kind of synapse is this? This is adrenergic, right? That's an adrenergic synapse. And then how do you do the effect? The effect of either can be either excitatory or inhibitory, because it really depends on what? What does it depend on? Yes. Depends on the receptor. And really to predict, I always think about resource allocation to predict. So I think about resource allocation, which is, can you afford it? Would it help? Okay, so now we back up a little bit and we look at this as a big picture again, and we figure out how the heck you can use this terminology and the fact that the concepts stick long before the terms do, okay? So what we're going to do is we're going to talk about this terminology and practice it a little bit and see if you guys can start to get to the point that you're answering them correctly. So the wording is where people get really tripped up. So I want you to think about this, and I want you to, I'm going to ask you a couple questions. What is the effect of parasympathetic, postganglionic, acetylcholine on cardiac muscle? Your two choices are excitation or inhibition. Inhibition, good. What is the effect of sympathetic, preganglionic acetylcholine? Excitation, exactly, right? What is the effect of parasympathetic, postganglionic acetylcholine on the smooth muscle in the wall of the uterus? Excitation, you got it? So do you see that there's like a lot of words and there's a lot of information in there, but the concepts are not very difficult? Let's see if you can do one that makes you sort of put together the table too. What is the effect of sympathetic, postganglionic norepinephrine on the smooth muscle in the wall of the bronchioles? Sympathetic, postganglionic norepinephrine, a smooth muscle in the wall of the bronchioles. It's inhibition to allow for dilation, right? So do you see that when you put it all together, it's a very wordy, very simple story, right? The logic is hard. The logic is easy and the words are hard. That's what I think. The logic is easy and the words are hard. Is that starting to make sense to you? Okay. It's not your favorite thing and it's not going to be your favorite thing. It's like the only thing that people like less than this lecture is cellular respiration. But the problem is that this actually, you can see that this is going to come back. When we start talking about cardiac muscle contraction, you're going to have to use this terminology again. When we talk about respiratory control, you're going to have to use this terminology again. Yeah? So you really can't avoid it. So might as well get it out of the way now. Okay, so last thing is to talk about the like fun things, like the functionality of how this like ends up playing like in CSI or whatever. So a lot of times what will happen is people will do like a polygraph test or a lie detector test. And a lot of what's happening during the lie detector test is based on this physiology. Because when you're doing a polygraph test or a lie detector test, remember in this country, they're not admissible in court. So primarily what they're used for is for people to exonerate themselves. But even if, for instance, it looked like you were guilty of cutting her up and sticking her in the trunk, they can't actually use it in court. So what's going to happen when they hook you up to a polygraph test is, let's say that somebody thought somebody murdered somebody, and you're gonna say that you will sit for a polygraph test. And if you have a really good polygraph examiner, this is generally how it's going to go. They're going to wire you up to measure a couple of physiological variables. One of them is obviously heart rate. Your heart rate tends to increase with the sympathetic stimulation. And then they also measure the electrical conductivity of the skin, because when you sweat, which is typically associated with the increase in body temperature of sympathetic stimulation, it increases the conductivity of your skin. They can also measure your respiratory rate. There are some that actually measure salivary gland secretion as well. So what's going to happen is they're going to hook you up and then they're going to start asking you some baseline questions, because generally you're probably going to be a little stressed out because somebody thinks you murdered somebody, right? And they're wiring you up to something. So they bring you in and they ask you, what do you have for breakfast? What's your middle name? What's your favorite color? Where do you work? And they're getting this baseline. And then they'll start like sprinkling questions in that are related. Do you know her? Did you buy this weapon? Is this your car? Did you kill her? Is she in your trunk? Right? And you sprinkle it in with these other things. And then if you have a good polygraph examiner, what you end up getting is a spike of sympathetic activity related to a certain line of questioning. And so all you can figure out in that situation is that I can't say that you didn't do it, right? I can say that you're still included as a possible suspect. However, if you know that you didn't do it, it's not going to hurt you to take that test, right? Like I tested them every way to Sunday, and either they're a psychopath Fascinatingly, psychopaths do not respond to polygraph tests in the same way because sort of one of the definitions of psychopathy or sociopathy is that you really do not feel the social tension of what other people think of you. So if I don't care whether you think that I killed her, and I don't really even feel particularly guilty about killing her, then I don't have a massive sympathetic nervous system response, which is one of the reasons that Ted Bundy got away with several polygraph tests before he actually ever admitted to killing all of the people that he killed. He was a like stone cold psychopath. Now, not all psychopaths are killers. Some of them are just like CEOs of major companies. So what about those situations in which, and I'm serious, like there is a completely disproportionate amount of psychopaths in like high level. Like just look at our presidential candidates. That's all I have to say right now. How is this related to like those legendary people who can like get through a polygraph test? Is it possible? Is it a myth? And the answer is it's usually a myth, but it is possible. And so what can happen is, let's say I'm a high-level CIA operative or I'm a Jason Bourne, right? And I know that I might be hooked up to a polygraph. You can train your responses with a lot of time. You can train your responses. You can look at the biofeedback that happens when you're in those stressful situations, and you can work on like what you're really doing is trying to train your cerebral cortex to override your hypothalamus activity whenever you have these certain inputs. And with a lot of time, you can actually make that occur. And so CIA operatives and some yogis and some martial artists have the capacity to do it, and most of us don't because we have other hobbies, right? We have like reality television that won't watch itself, champagne that's not going to drink itself, right? Those kinds of things. So it is possible. It's just not very likely, and I've never known anyone who could do it. Do you guys know anybody who does polygraph tests? Damn it, I ask every single semester because I totally want to get wired up to one of those things. I want to see, like, how good a liar am I? I don't think I'm very good, although when I was a teenager, I thought I was amazing. I'm pretty sure my parents just weren't paying attention. Okay, so let's see. We will stop there. We'll start on the endocrine system next time. Oh, let me tell you something about the endocrine system, though. I am going to tell you just with the last 2 minutes, a couple of things, and that is this. Okay. With the endocrine system, first off, I'm going to reorganize your endocrine notes. I apologize if you already printed them out. I'm not adding any more information. I just figured out a better presentation, so I'm going to like tweak them a little bit. Hopefully they'll be shorter, but I can't guarantee. They won't have any more information in them, even if they're longer. So wait, and I will redo that one. And then with the endocrine system, you are also going to have a table where you can organize like where the hormones come from, where they go to, disorders, chemistry. I'm going to work on those over the weekend too. So will you just do me a favor and wait until it says it's updated before you download those? And then we will start working on them on Monday. Now, I know that you may be tempted to miss class on Monday because you have a test on Wednesday and it's not on that test material. That would be a super duper bad plan for endocrine system. Okay, endocrine, I think you need me or an instructor anyway with endocrine more than with lots of body systems. Okay, so I would be here if I were you. Okay, have a good weekend. The test goes through autonomic. So the test started with synapses, muscles, and autonomic, not endocrine. Right? Synapses, muscles, autonomic. Because we went through membrane potentials on the last one. So these 4 were on the last one. Synapse, muscles, autonomic on the next one. Good? I haven't written yet. Probably about the same as it was last time. Did you feel any better after sitting through it, after listening to it?
Yeah.
You did? Okay, good. Right. Because you're a visual person. Mm-hmm.